विज्ञान की अनकही कहानियां, आइए हम सुने विज्ञान की अनसुनी कहानियां, एक नई दुनिया की आवाज आपकी आवाज, विज्ञान की आवाज, की आवाज.
Celebrating Azadi Ka Amrit Mahotsav, we bring you a podcast series on STEM popularization, science, technology, engineering, mathematics and medicine, supported and catalyzed by the National Council for Science and Technology Communication, Government of India. This program is produced by Media for Community Foundation. Creative content and technical support is given by Anuradha Kunte, Yeralawani Community Radio, Sangli, Maharashtra.
एक नई दुनिया की आवाज आपकी आवाज, विज्ञान की आवाज, की आवाज, की आवाज.
Welcome friends, you are listening to STEM Popularization Podcast. Friends, we are little bit aware about fungi, that they are responsible for breaking down organic matter and releasing carbon, oxygen, nitrogen and phosphorus into soil and atmosphere. Also they are essential in making bread, wine, beer and certain cheeses. Today we are putting more light on biodiversity of fungi. Let's meet Dr. Rajeshkumar KC. Dr. Rajeshkumar KC is scientist, National Fungal Culture Collection of India. It's a part of biodiversity and phylology of fungal group at Agarkar Research Institute, Pune. He is mycologist by profession and completed his PhD from Forest Research Institute, Dehradun. He worked on plant pathogen fungal diversity in Western Ghats of Kerala. Here, Dr. Rajesh is explaining about his research in biodiversity of fungal group. So, let's meet Dr. Rajeshkumar KC and know about his journey from research at Kerala till mycologist at Agarkar Research Institute, Pune, and also about his research in his own voice.
We have surveyed forest areas of Kerala and identified these plant pathogenic fungi and made a list of these fungi from the natural forest. After completing my PhD, I did a postdoctoral research with VKA, UK. Previously it was known as International Mycological Institution. We worked on biocontrol of weeds from the Himalayas and identify potential pathogens against weeds, noxious weeds, which is noxious weeds in England. Actually, these plants were taken earlier during Victorian periods from India. So we wanted to find some natural enemies for these plants. So along with British scientists from international mycological institutions, we have identified potential rest pathogens, which is only killing that particular plant in India. This plant is not a weed in India, but it's a weed in UK because these natural enemies were not there. After completing that project, I have joined in National Fungal Culture Collection of India as a scientist B, junior scientist, and continued my research in more focusing on the mycology aspects, like as a fungal taxonomist. Then for last 15 years, I've been working here as a mycologist, dealing with especially in the taxonomy of Ascomycota group, that means true fungi group. As I have mentioned, we have majorly four missions. First one is ex situ conservation of the fungal germplasm of India. That means we collect fungal from across the India, from the natural forest, other sources. And also researchers contact National Fungal Culture Collection for identification and validation of these species. And we conserve these fungi for long term and make a bank for these collections. And during that process, we do the accurate identification of these species based on morphology and modern tools. Modern tools means currently DNA-based identification is considered to be the authentic way of validating the species. So we do the molecular sequencing for which is called multi-gene sequencing also. Based on that, we can authenticate these species because accurate identification of species is important for any research, especially of fungi, which any research, for applied research, if you are taking a wrong fungus, the whole result will be get affected. For example, if you want to identify a plant pathogen, which is a noxious pathogen, you have to identify it accurately. In order to do that, we are using multiple tools. That is considered to be polyphasic taxonomic approach, which is a most modern concept in the world, where we identify this fungi through morphological aspects, molecular multigene sequencing, and metabolite profiling. In a compilation of these, we identify and also we place these fungi in the fungal tree of life. That means phylogeny-based, classification, where these exact fungi is falling in the classifications. So that is the basic aspects of NFCCA where we are doing. Other than this identification and germplasm collections, we are giving training to youngsters from across India. We train PhD students, researchers, scientists. This is an integral part of workshop and training programs, an integral part of National Fungal Culture Collections, and we are also contributing to that. Additionally, we are doing research and development. Research and development is based on the interest of scientists in the facility. So currently, I am focusing my research on three aspects. First one is I am working on diversity of lichenized fungi from the Western Ghats. And the second project is focusing on lignin degrading fungi from the natural forest of the Western Ghats. And the third project is focusing on the phosphate degrading species of Aspergillus niger. So we have developed expertise in Aspergillus taxonomy based on molecular and morphological taxonomy, and we have had projects from DST SERP earlier. So we have assessed the diversity of Aspergillus and Penicillium in India. Based on that, we have constructed this project on phosphate solubilizing fungi, and currently 3 PhD students are working. Actually, most of my research is well aligned with the national policy of the government and even DST. We have projects like National Action Plan for Climate Change. So actually, in fact, what is happening? While assessing the diversity of lichens in disturbed and undisturbed area, that means pristine area. So we are going to the natural forest and understanding the disturbances through transportations, road, tourism, other places, and we are collecting lichens, especially a primitive group of lichens which are called Questros lichens. We are working on a family which is called Graphidaceae. Actually, you must have heard about lichens, which is a composite organism which is made up of algae and fungi. So they thrive in a specific microhabitat which needs special associations. So we try to unravel this understanding, like symbiosis, which is existing through studying the diversity of fungi through molecular tools, as well as understanding which kind of algae is present. So these lichens are pollution indicator species. So when heavy metal pollution happen in a particular area, there will be depletion of the diversity of the species. So when we compare the disturbed and pristine habitat, we can directly or indirectly assess the diversity of lichens in that area, as well as we can predict whether heavy metal pollution affect the diversity. That is directly related to the National Action Plan for Climate Change, where we generate knowledge for climate change, as well as the Green India Mission, where the health of the forest will be assessed, as well as how to improve the quality of the forest. So in order to assess the quality of the forest, we need to know whether it is a healthy forest or degraded forest. So this project is in line with that. The second project is more towards is national bioenergy programs. We need biological resources like fungal resources for production of biological, like bioenergy, in future energy. There, lignin degrading fungi are most important because in natural forest we have a lot of biomasses available. So under the biomass programs, we have to utilize this biomass for future bioenergy production. So how to do that? Currently, this lignin is degraded to mostly through chemical treatments, and which is considered to be very costly. So we wanted to identify potential fungal biological resources from the natural forest. So one of my students is surveying in the pristine natural forest where the bioconversion rate of lignin or natural organic this polymer is very high. So when we research on such area, we collect specific fungi which are unique to that ecosystem. As we are aware that 1/3 of the fungal diversity exists in India, and India is considered to be a biodiversity hotspot. So these biodiversity hotspots are rich and diverse, the different groups of fungi, and many of the fungi are very unique to India. They are only found in India. So collecting these fungi, identifying and publishing through the most modern tools, which will give the biodiversity aspects also. Also, we can explore this exclusive specific germplasm, which is present in India, for targeting this kind of research. So in that research, we are planning to have a germplasm from India, and these groups are comparatively less studied for degradation of lignin. We are assessing the diversity, placing them in the molecular tools in the fungal tree of life. That means most modern group of sets of rules which we follow for characterization of fungi. And the third project which I am dealing with is phosphate-related, phosphate degrading, like solubilizing fungi.
You are listening to STEM Popularization Podcast. We heard about types of fungi and the identification of varieties of the same. The biodiversity that is being researched here in India. So we would like to know furthermore on the use of this biodiversity of fungi research carried out by Dr. Rajeshkumar on the lives of common man. So please, Doctor, add more about the research stage and where our country stands globally in this kind of research.
Actually, under National Mission for Sustainable Agriculture, there is program for soil health management and sustainable agriculture programs. So under this, you can understand that this soil is continuously under cultivation and they are continuously using chemical fertilizers. So as you have heard that Sadhguru has conducted a international tour for soil health, and we have to protect the soil. So this is in line with the mission of National Mission of Sustainable Agriculture, where we can utilize this fungal germplasm for topping the decay, improvising the health of soil. And when we identify potential fungal strains, for example, Aspergillus niger is considered to be most economically, important group of fungi. They are used widely in industrial application of production of acids, and as well as in various applications. So when we identify this black aspergillus, it is very daunting task because of, it's considered to be a nightmare for taxonomy to separate species. For that, we are utilizing the most modern tools and techniques, which is authenticated by international agencies in fungal classification. And we do that. We have collected at least 10 to 15 different species of Aspergillus from India. Then we test its solubilization potential of phosphate. Then we identify some super producer where phosphate is highly solubilized. What is the real problem is when we add this NPK into the soil, most of this phosphate gets mineralized. So it is not available for the plants. It's only like 20 to 30% is available. Rest of the leach out to the water and it just get mineralized in the soil and it becomes stable. So we need these biofertilizers development for bringing demineralization and making these phosphates available for the plants. As well as during this process, we can also develop biofertilizers. The biofertilizers is a natural approach for, ways to recover the health of soil. So that is the third program which I am working. India is in par with the international research which are going on in fungal taxonomy. As National Fungal Culture Collection of India, with the establishment from last 15 years, we have contributed immensely to enhance the quality of research in India, and we are one of the largest culture collection in India, and across the globe under the World Data Center of Microorganisms, we are a part of global catalog of microorganisms, and we are following almost all authentic procedures which are needed for the characterization of fungi. And our team also contributing to the applied research. My coordinator is exclusively working in that. When we compare to the countries like such as Europe and UK were the pioneers in taxonomy of fungi and validation. Later on, Europe take over that, and they have the largest fungal culture collections, and where they have standardized almost all protocols for international level, and they are the leading countries. Currently, China is also one of the major country investing a lot in fungal research, especially countries like Thailand. There were one institution like Marfaling University. They are publishing at least hundreds of publications from the Asia. So they are currently leaders in fungal taxonomic publications. But as far as India is concerned, yes, we are up to the par with the international countries, and we are also leading countries among Asia. Actually, we are publishing in almost every journal across the globe, and among the leading journals we have published in Fungal Diversity, which is considered to be the meritorious journal. We have published in Studies in Mycology, which is from Europe. Personally, I have published around 60 international publications with almost every journal from across the globe. And we are also collaborating in some publication with European, Canadian scientists from Thailand in order to the quality work for India. We are working in various aspects of fungal taxonomy and its application. Yes, my department is associated with different groups. And yes, recently also we have taken a patent for a molecule which is identified as an anti-cancer agent. So we are in collaboration with different institutions, and obviously we'll be taking up all these, like for example, this bioenergy related research into industrialization. That is the long-term goal for us. Even for phosphate solubilization, we need to help farmers of our country. So we are taking up that research into that direction, and we are also keen to work more towards climate change studies. I'd like to thank Department of Science and Technology for giving me this opportunity and talking about my research area and National Fungal Culture Collection of India and our activities. I also thank Garka Research Institute.
Thank you, Dr. Rajeshkumar, for being with us on the STEM Popularization Podcast.
आइए हम सुने विज्ञान की अनकही कहानियाँ। आइए हम सुने विज्ञान की अनसुनी कहानियाँ। एक नई दुनिया की आवाज़ आपकी आवाज़, विज्ञान की आवाज़, की आवाज़।
Celebrating Azadi Ka Amrit Mahotsav, we bring you a podcast series on STEM popularization, science, technology, engineering, mathematics and medicine, supported and catalyzed by the National Council for Science and Technology Communication, Government of India. This program is produced by Media for Community Foundation. Creative content and technical support is given by Anuradha Kunte, Yeralavani Community Radio, Sangli, Maharashtra.